Welcome. This is the third session for today. My name is Andreas Tamaris. I'm the president-elect for the Greater New York ACSM chapter, and it's so good to see you here. I'm here to present two great friends. One is coming from Spain, and the other is coming from Greece. So, Dr. Sergi Garcia-Retortillo studied exercise science and physical therapy at the University of Barcelona, where he obtained his master's degree in physical activity and health. In 2006, he graduated with a PhD in exercise science. Currently, he serves as visiting researcher at the Keck Laboratory for Network Physiology in the Department of Physics in Boston University. As for his scientific experience, his research program is based on the new field of network physiology of exercise, which investigates basic laws of interaction and principles of coordination and integration among diverse physiological systems during exercise. They have developed a novel variable, cardiorespiratory coordination, which provides information about the coordinated activity between cardiovascular and respiratory systems during cardiorespiratory exercise testing. Currently, he is investigating intermuscular and inter-organ network interactions during exercise. His medium-term aim is to develop unique fitness assessment systems using novel network-based biomarkers with the potential to open new and exciting horizons in exercise testing, exercise science, and other health-related fields. The most relevant network-based biomarkers of his research agenda are cardiorespiratory coordination, intermuscular coordination, and corticomuscular coordination. At present, he serves as guest associate editor for Frontiers in Network Physiology. Further, he has extensive experience in the fitness and wellness industry as a strength and conditioning coach and physical therapist, and has trained a wide range of athletes and patients for over a decade. And for our second speaker joining us today is my old roommate from Baylor University, Dr. Zacharias Papadakis from Greece. Holds a BSc, a Bachelor's of the Physical Education and Sport Exercise Science program at the University of Thessaloniki, Greece. He obtained his Master's in Exercise Physiology from the Faculty of Health and Wellbeing at Sheffield Hallam University in the UK. He completed his PhD at Baylor University in Waco, Texas in kinesiology, exercise, nutrition, and health promotion. Before joining Barry University, Department of Sport and Exercise Science in 2018 as an assistant professor, director of the Human Performance Laboratory, and director of both the undergrad and the grad exercise physiology and human performance and wellness programs, respectively. He worked as an assistant professor in teaching at Rice University in Houston, Texas. His research interests are aimed at understanding how the human body responds to various exercise, nutritional, and pharmaceutical stimuli, and how these responses influence vascular reactivity, inflammatory, and metabolic processes in humans under both the traditional exercise physiology and network physiology of exercise concepts. He teaches in the area of exercise physiology, clinical exercise physiology, strength and conditioning, research, designs, and stats. He's also an ACSM certified exercise physiologist. Let's welcome both presenters.
Okay, good morning, everybody. Thank you so much, Andreas, for your kind introduction, for your invitation. It's a real pleasure for us being here. So our talk today will have, let's say, two separate parts. We will first introduce the concept of network physiology, the new field of network physiology and network physiology of exercise. We will discuss several data analysis methodologies to infer organ interactions. And then we will present the human physiolome, which is a new atlas of organ communication, of organ interactions. And finally, we will present some of our new network-based biomarkers. We are developing now these markers. And let's say these markers are able to quantify how different muscles and even how different organs coordinate among them during exercise. Okay? So we will present intermuscular coordination, we will present cardiorespiratory coordination, and among others. So let's start with the introduction of network physiology and network physiology of exercise. So as you know, the human organism is composed of multiple organ systems, right? So each system has its own complex structural organization. And normally these systems present a kind of nonlinear behavior. So in order to assess and to identify health status, medicine and physiology normally investigates organ systems in a separate way, separately, right? For instance, neurologists investigate the brain and they utilize EEG, MRI, etc., right? So pulmonologists, they utilize respiratory patterns, cardiologists, ECG, echography, etc. So, and what about us? What about exercise physiologists? What about strength and conditioning coaches? What about physical therapists? So we do exactly the same. We utilize several variables. Of course, they are very useful, but these variables mainly provide information regarding one system, right? VO2 max cardiovascular, power or muscle, heart rate, the heart, which is okay. Actually, human physiology and exercise physiology during the last 20, 30 years has mainly focused on the integrative, let's say, vertical integration in physiology. So what does it mean, vertical integration? If I want to investigate the brain, I will take the brain and I will go deep and deep and deep and deep, and I will investigate the effect of a given training program on a given neuron, on a given protein, whatever. Same for the muscle, same for the heart. This is great, and thanks to this approach, science has evolved tremendously during the last years, right? However, and I think you will all agree with me, the human organism is an integrated network, where multi-component organ systems continuously interact among them at several time scales in order to optimize and coordinate function and to generate health. We know that coordinated interactions of organ systems are essential, are very important, are of key relevance to generate and to maintain health. So we also know that altering or disrupting organ communications can be dangerous, can lead to several health problems, right? So despite the importance of organ interactions, I think we know almost nothing about organ interactions. We know that organs communicate among them. We know that muscles talk among them, but we don't know nothing. I mean, we know nothing about it. So this leads me to introduce the horizontal integration in physiology. So horizontal integration refers to, to investigate how several organs coordinate among them during in a given level of integration, right? Network physiology, this new field in human physiology, can provide information with respect to horizontal integration. Actually, in other words, network physiology, this new field in human physiology, which was introduced by Professor Ivanov 10 years ago from Boston University and Harvard Medical School, network physiology puts its research efforts on understanding how different organ systems, the brain, the heart, lungs, muscles, communicate among them, and track how this coordination leads to different physiological states. Let's say wake, fatigue, disease, whatever. Okay? So more specifically, we have recently introduced like a subfield within network physiology, which is network physiology of exercise. Okay? Network physiology of exercise investigates how different muscles, how even different organs coordinate among them during exercise, and investigate how this coordination among organs, among muscles, change in response to fatigue, in response to different training interventions. Okay? So, and now you can say, okay, Sergi, that's great. You have been talking for 5 minutes about organ interactions. About how do you measure, how do you quantify organ interactions, right? Well, first, we need high-frequency recordings. High-frequency recordings means time series of many data points. long time series. For instance, if I want to investigate the brain, I will need EEG. For the muscle, I will need EMG. For the heart, I will need EKG, high frequency recordings. So once I have two signals from two different physiological systems, the brain and the heart, for instance, or the brain and the muscle, I will need a given data analysis methodology to quantify the coupling, the coordination between these two signals. So there are like many different computational tools to, let's say, quantify such couplings. We will use one technique or another depending on the nature of the data or depending on the type of exercise protocol, right? For instance, here you can see the time delay stability. Time delay stability is one of the most utilized data analysis techniques to quantify organ interactions in network physiology. Here you can see an example. I will just talk about it very, superficially. You have four physiological signals: EEG, the brain, heart, respiration, and eye. You can see here the vertical red line indicates several bursts. Each signal has several bursts of activation, and these bursts can be observed in all the signals. However, these bursts are not perfectly synchronized in time. They have like a delay of several milliseconds, right? So time delay stability quantifies the stability of this delay across the time series. Okay? So depending on the stability of this delay during the time series, we can say that these signals are coupled or not. Okay, we can, let's say, identify the level of coordination among these signals. Okay? It's a bit complicated, algorithm. You can, if you are interested, you can check these papers which are already published. We have many other methods. For instance, we have also the cross correlations on spectral power time series. I will give you an example later. We have principal components analysis, and Zach will show you later. We have phase synchronization. We have a lot of them. Again, we will utilize one or another depending on the nature of the data. Okay, so, and now you can say, okay, what do you want to do with this? So our vision for a network physiology is the human physiolome, right? So now we have atlas of human anatomy. These are topological atlas. We have the biceps here, the brain here, the lung, whatever. This is good. So now we are developing, together with Zach and our group and other people, what is called the human physiolome. The human physiolome is an atlas of dynamic interactions of organ systems. This is crazy. Of course, you can imagine. You have like hundreds and thousands of physiological maps. So this information here can complement very well the topological information. We need to complement this, right? So this is our vision. And this is a very serious work because, the human body produces a gigantic amount of data and information. It's insane. Imagine if we collect simultaneously high frequency recordings. Remember, we need high frequency recordings from the brain, the heart, body temperature, muscles. We can get tremendous, huge amount of data. Okay? This is real big data. So that's why we need to develop new computational tools, new systems to process data. And we, as exercise and health scientists, we need to improve our programming skills, our coding skills, because there is no way to process such amount of data using Microsoft Excel or using SPSS. We need something powerful, okay? Such artificial intelligence, as Ali presented before. There's a very nice connection here. So now, let's say we have our approach, the umbrella of our work, which is network physiology, the human physiolome, but how we can utilize this information in exercise and health science. So the most imminent practical implications of this human physiolome, of this, let's say, data analysis methodologies is exercise testing. So actually, we are developing network-based biomarkers. So again, network-based biomarkers are new markers able to quantify with numbers, quantify how different muscles, how different organs coordinate among them during exercise. and can track how this coordination changes in response to acute fatigue and to different training programs. This is very important. So now let's move to the second part of this talk today. Now we are going to present some of our biomarkers. Again, we are developing this, so we are on our first steps, but you can see at least our starting point. So our first biomarker is intermuscular coordination. Okay, intermuscular coordination obviously is able to quantify how different muscles coordinate among them during a given task: squat, push-up, whatever. So here I will present you one of our experiments, so you can, at least have the big picture of our work, and you can see the potential of these markers. So here our participants performed 3 sets of body weight squats without additional weight, right? They performed 3 sets until real exhaustion. So this was a very tough protocol. Imagine 3 minutes squatting, it's kind of difficult. So during the entire protocol, we collected EMG data from 4 different muscles: right and left vastus lateralis, and right and left erector spinae. Okay? So what did we find here? Let me show you this figure. So here you can see 3 networks. Okay, so we have one network for each set of squats: exercise 1, exercise 2, and exercise 3. So let me try to help you to understand this, because it is important. Each network has 4 semicircles, you see? So each semicircle corresponds to a given muscle. We have the back left. We write here back instead of erector spinae just to make it easier. Back left, back right, leg left, and leg right, the 4 muscles. So within each semicircle, for instance here, we see like several nodes. So these nodes represent different frequency bands within this muscle, in this case within the leg left, the left vastus lateralis. So these frequency bands can at some point represent the activation of different type of muscle fibers. Okay, because this is a frequency analysis. Maybe later we can discuss more details about the analysis. So F1, F2, F3, these first frequency bands can sort of represent the activation of slow muscle fibers of the leg left. F5, 6, 7, 8 can also be associated with the behavior of the activation. Yeah. Yeah, sorry, set 1, set 2, and set 3. Sorry, yes, it's set 1, 2, and 3. Yeah, thank you for the point. So you can interrupt me whatever you want, no problem. So F5, 6, 7, 8 could be associated with the behavior of or activation of intermediate muscle fibers within the left vastus lateralis. And finally, 8, 9, 10 with the fast muscle fibers. Okay, and the same for all the muscles. So you got this? Great. Then we have the links, the lines. The links indicates the coordination or represents the coordination between 2 different frequency bands, between 2 different muscles. For instance, this line here, this link, indicates how is the coordination between low frequency bands, or let's say slow muscle fibers in the leg left and slow muscle fibers in the leg right. Okay, and all the possible combinations. Here we have 600 links. 600 links within each network. Okay, so then finally the color of the lines, the color of the links indicates the strength of the coupling, the strength of the coordination. So as you see in the legend, strong links are magenta. Magenta indicates strong links, strong synchronization between the 2 muscles. Blue links are intermediate links, a bit weaker. Green are, let's say, weak links, and gray are very weak links. So now, what can we see here? If you compare, if we take, let's say, for instance, the main muscle pairs, which are leg to leg and back to back, back to back and leg to leg. These are the main muscle pairs. If we compare exercise 1 or set 1 with exercise 3, we see that, I mean, the magenta links almost disappear. We only have a few magenta links. We mostly have blue and some of green links. This indicates that there is that fatigue generates an important generates a reduction on the levels of coordination between these different pairs of muscles, between back to back and leg to leg. This is clear, right? So fatigue generates a reduction of coordination. Okay. But then what happens with the cross frequency, cross muscle communication? If I take this diagonal here, back left with back right, what happens? If I compare set 1 with set 2, I can see that in set 2, look, there are more links, there are more lines, and more green lines. What does it mean? This is a clear mechanism to compensate the elevated levels of fatigue on the main muscle pairs, back to back and leg to leg. Okay? So you can say, okay, Sergi, I mean, this is not shocking. We already knew this. We already knew that, you know, the neuromuscular system reorganizes with fatigue. I mean, this makes sense. But what's the point here? The point is that we are able to quantify this. Each line is a number. That's the case, that's the point of this. Each link is a number, and behind this number there is a very, let's say, complex algorithm. Okay? But what is the real point of this? We replicated these protocols with different basic movements. We replicated these protocols with squats, pulls, and push-ups. I mean, these are the basic movements in the human organism, right? I push, I pull, and then I go down. That's it. Then I have combinations. So the point is, it turned that we believe that we uncovered the first physiological laws of intermuscular coordination during basic movements. What does it mean? For instance, coordination between two muscles will depend on, first, the type of the muscle, and second, the role of the muscle, of this muscle plays during the task. So we found like 2-3 different patterns. The first one is this one. So if we have two, let's say, two muscles which are the same, for instance, leg to leg or back to back when I am squatting, or chest to chest, back to back when I am doing push-ups, or lat to lat when I am pulling, right? This is the way in which these muscles will communicate. It's like a universal law, or can be a universal law, can be. We will figure out in the next month. So again, here you can see like three networks, one network for each, let's say, set of exercise. And you have two semicircles. Here we have only two muscles. Yeah, in this case, it's leg to leg, but it could be back to back or chest to chest when I am pushing up, or lat to lat when I am pulling. Okay. And again, you see the frequency bands, you see the links, you see everything. And then the bar chart to the right, quantifies the network. So as you can see here, there are like several groups of bars. Now we don't care about it now. Just have a look at the shape of the different bars. It's like there is a particular shape. This shape indicates that each type of muscle fiber within, in this case, the leg left, communicates with all types of muscle fibers in the leg right, and vice versa. There is like a spread communication. Okay? What happens with fatigue? With fatigue, you see this shape changes a bit. It's the same shape, but it's like there is more spread. This means that there is a cut on the frequency, on the cross frequency communication. This means that slow muscle fibers with fatigue only talk with slow muscle fibers, and fast to fast, intermediate to intermediate. This is a cutoff. This is a clear effect of fatigue, and we will see this leg to leg, back to back when I am squatting, chest to chest when I am pushing, lat to lat when I am pulling. It's like a more or less consistent way of communicating. Okay. So what else? What happens if I take different muscles? If I investigate coordination between two muscles which are different. For instance, leg to back when I am squatting, or chest to triceps when I am pushing, or lats to biceps when I am pulling. Look, the things are different, right? The networks are different. You know, the bar charts, the shape of the bars are different. This means that since the very beginning, when two different muscles, let's say, talk between them, there is an initial cut of cross-frequency communication. I mean that since the very beginning, slow muscle fibers only talk with slow, intermediate with intermediate, and fast with fast. That's it. I mean, it makes sense. We have two different muscles, so different histochemical characteristics, different role. So therefore, the initial profile is different, and the response to fatigue is also different. It's like the second pattern. Okay. So now we are on the process to replicate these protocols on larger populations, because we want to obtain reference profiles. Reference profile. We have reference values for VO2 max, heart rate. For all the tests we utilize, we need reference profiles for this. We are trying to develop this, because at the end we want strength and conditioning coaches, physical therapists, exercise physiologists. We want these people to utilize these markers to assess clients, to assess athletes, to assess, people in general. Okay. Good. So let's close this intermuscular coordination now. We can further discuss later if you want. This is like the big picture. And now we will present you some of our second biomarker that we are developing now, and Zach will continue. So our second biomarker is cardiorespiratory coordination. This marker can quantify how the respiratory and the cardiovascular systems coordinate between them during a cardiorespiratory exercise test with gas analysis. Okay. So since the type of data is different, we utilize different data analysis methodologies to quantify the coordination. In this case, we utilize what is called the principal components analysis. This is a more or less easy way. It's not complicated. So principal components analysis, in a nutshell, very briefly summarized, is able to quantify the degree of covariation among a set of cardiorespiratory variables during a maximal exercise test. So again, it can quantify the covariation between several respiratory variables such as ventilation, expired fraction of oxygen, CO2, heart rate, blood pressure, etc. And this principal components analysis provides us with a number, with a value, which are the number of principal components. Now it doesn't matter. But as a general rule, just keep this in mind because Zach will continue now. We know that if we find in a given participant during a given test a high number of principal components, okay, high number of principal components, this will mean that the degree of cardiorespiratory coordination is low. The degree in which the respiratory and the cardiovascular system communicate is not very efficient, as a general rule. On the other way, if we find like a low number or reduced number of principal components, one mainly, this will mean that the level of cardiorespiratory coordination of this person during a given test is more or less high. Okay, this is like the general rule. So now Zach will continue and will show you some results obtaining elite rowers, and he will finish. So
thank you so much.
All yours.
Do you use microphone or no? Yeah. Okay.
Ready?
You can use that, yeah. Can you hear me well? All good? Okay, so I hope that we didn't lose you so far with all these like concepts, but oops. Okay, so this like work that Sergi has done, and now we try to take this the next step to be applied, right? Because it's nice to have a theory, it's nice to hear all these concepts, but then we have to take that concept and put it in the actual like field of exercise science. So what we did, I work in a university that I have access to athletics. We run tests every year to basketball, volleyball team, rowers, golf, tennis, name it. I have access because I work in Division II. So it's easy for me to get these athletes in. And
part of this was we had these rowers that they're going to do a VO2 max test. So I want to see how this new network physiology concept can actually be applicable to the coach. Because coach, they take these VO2 max results, okay, just for the cardiovascular system, and they classify their athletes, okay, on the boat. And I want to provide like an extra level of, let's say, assurance, an extra level of probably information that I can get to justify who is sitting with who, okay, who is pulling the rope better than the others, okay. Also in this like group, we had one Olympian from Argentina. So it was good for us that we were able to compare students that they are not all Americans. Students actually are Olympians, and students actually try to make it the team. Okay, and this is something we actually published as an abstract in the and present this in the Texas ACSM, and right now we're writing the manuscript. But what we found, as Sergi discussed earlier, is like this coordination. Okay. And I'm not going to say too much on the graphs. I'm going to just show you how the level, like these lines on the top and the bottom, they change, and they signify the components, okay, of the athletes that actually completed this test. Okay, as you can see, we have here like on the right side the number of personal components, and you can see that we have these like athletes that some they have one or two. So if you recall from the previous presentation, one means what? Good coordination or bad? Good, right? Two means bad. Okay, so the less that you have, the better it is. So right now we have here one athlete. Let me find who that might be. It's Alejandra. It's over here. Okay, so Alejandra AA, okay, is the Olympian, and as you can see, has two. So based on the concept of the network physiology, Alejandra has a great room for improvement because something in her system, some of the training program that she does, okay, with a coach, doesn't actually work for her well, because the coaches, what they do, even though we like exercise medicine, exercise should be prescribed individually, coaches, they have one program, okay? They train the athletes, student athletes, the same way. So here, okay, we can see that we have this freshman, okay, that has one. So that freshman is the best athlete, okay, in that perspective, that neurophysiology. And since I have like access to other data, that actually this person is not the best in the team. So that person can actually coordinate very well. Maybe that athlete has reached the maximum level based again on this neurophysiology of exercise perspective. The point right now that I was trying to make the coach is that Alejandra, okay, since she's Olympian, and based on that perspective, even though her VO2 max is not the best, it's lower than the best, we have other athletes that they have one principal component, but their VO2 max is the highest, she needs to be trained differently. We need to find, okay, what's the case for that athlete that based on this concept, based on this statistical analysis, her cardiorespiratory coordination is less, has like two components. The other thing that I would like to do, because I do cardiovascular stuff, I would try to take things from the real life and see how I can actually utilize them for us. So this is a study that we did, and we actually tried to see how sleep, exercise, and bad diet interact. We know that if you don't sleep well, okay, your health goes down. We know that if you eat like bad things, burgers, prepackaged food, cholesterol, everything goes up, so that's bad for your diet, bad for your health. But we know exercise is beneficial, right? Exercise is medicine. But we want to know what happens if all these things interact together. So we did this study, and we actually published like 3 papers with that one, and we examined a variety of cardiovascular markers, okay? We found at the end of the study that everything, okay, based on that design, exercise was cardioprotective. It didn't matter if you actually have been able to sleep 8 hours, that's the perfect like time frame, or maybe be under like sleep deprivation, because we did this like very intense, okay, protocol. They had to be in the lab for hours, okay? This is the time I was introduced to PlayStation because I had to keep my subjects somehow interesting and stay in the lab. And then they had to go home, they have to sleep for 3 hours, and then they have to return to do a series of blood work, running, okay, high intensity indoor exercise. And then after that, they have a very fat meal. This is probably what everybody does. I mean, I'm an avid like exerciser, so I work a lot, I sleep less, okay? I woke up in the morning, I exercise, but also I'm a foodie. I'm European, right? So then I have the thing that, oh yeah, I exercise. I can eat like right now, whatever I want. Let's have a pancake. But okay, does the exercise help me? Yes, no. So I want to test this. So we test this, and yes, exercise cardioprotective. It's fantastic. I was like, yeah, I'm a researcher, I'm a scientist. I find something good. Perfect. Let's keep it. Then we said, okay, those are the results. I have to speed up a little bit. Then we said, let's examine the same concept and a different perspective. Okay, let's try to examine this. Actually, something that was accepted for application in the frontiers of physiology. Let's examine the same thing, okay, under the network physiology of exercise. What might happen? Well, we're doing the analysis, and I was always thinking that, okay, exercise is going to be good. Exercise is going to be good. But again, based on this analysis, we found that exercise actually wasn't cardioprotective. Okay, the sleep deprivation in that case, in that perspective, it was actually like a strong stimulus for these bad habits. This is the first, okay, correlation map. So you can see we have a difference between the colors, okay, the negative links, and we assessed only, okay, just the LDL network, like the bad cholesterol, right, to see how it interacts. And you see here, okay, the exercise, okay, with the reference sleep, and then nothing much, right? And then we go to the other network, and you see all these interactions, all these nodes, all these like negative links. So something's happening, and probably it's because of this short sleep, because this one is reference sleep, 8 hours sleep and exercise, right? And then because I want to prove that exercise is beneficial, no matter what, if you sleep well or not, newsflash, no, it's not. So the point again is to create this new data set, these biomarkers, so you can utilize this to actually see, like look inside the box, see, think outside the box, and see how we can actually re-examine concepts that we've been taught, okay, in a different way. I'm not saying that the VO2 max concept is wrong, okay, the fatigue like models are wrong, but we may find different ways to clarify and identify these markers in the future with these novel techniques. One of the things that I have in my lab, and we heard like Dr. Bolani earlier discussing about how it should be like wearing like these things on the head and do a lecture. We try to use these like markers, okay, for esports. We have a study with esports, and we try to compare shooting athletes that actually shooting athletes. They play sports and they're esports, because I have a concept based on the traditional philosophical terminology that esport is not a sport, it's a gaming, okay? So these esport are not athletes, and they don't maybe deserve NCAA grant funding. So we try to compare this by having this eye-hand coordination. We have like a galvanic skin temperature, oxygen saturation, different markers, heart rate variability, and we have like these video games. They play, we collect like raw data, massive data, and then we try again to create, as Sergi said, the reference values, okay, to see how the shooting athletes, the real shooting athletes, they coordinate, they synchronize their eye movement coordination. Baseball, one of the best sports, right? Compared to, let's say, a video gamer. Another thing that we do is with the Curia, with the OREC over here, we're like in the talks, and we're like in very advanced talks, and probably going to give me some of their staff to do a series of studies in my lab. ORECO is a company based in London, from scientists, from researchers as us. Everybody works in the university, and this is their sidekick over here. So they just collect blood, and they analyze the blood for oxidative markers. Okay, everything regarding oxidation, you can get with a kit, like a glucose kit, very, very small kit, and then you upload this to your system, to your account, and then you get this type of, let's say again, graphs, okay? And ORECO says that everybody has a pattern, okay? A line that represents the health status and the unhealthy one. So they have a clientele in Europe, all like the major soccer teams, and they reach out to us over here, and we try to launch right now a study with University of Alabama and Clemson to test football players. They have done like a previous study with Oregon State with football team. Right now, they want to go with the best teams in the country, and we're like in the talks to actually take everything we discussed right now, the network physiology of exercise, and actually implement the biomarkers with this blood work. And how we do this, again, as I said, we need to have systems like the ADI instruments that can collect simultaneously for different places in our body, raw data in high frequency, and then we can have this vest that we can get the heart rate, temperature. We have this cup for the brain activity. Of course, the electrodes that we put in the chest for the EEG and ECG, and then we get signals like this. Okay, and again, we need to have great aggregation of data over here. We have to analyze this, and we need to be able to use this MATLAB, R, or whatever to be able to filter this out and get the signals that's most important for us in order to create these biomarkers. Because the end of the point is that we take this and then we put in a watch, and then you wear that, and then you get all this information, and then you can be alerted that, okay, you're healthy, you're tired, you need to get rest, you need to do your exercise, something like this. I hope I was right on time. I had to rush up a little bit. So we're ready for your questions.
Thanks, Sergi. Thanks, Zach. Really interesting. You keep talking about high frequency data. In case somebody doesn't understand, what do you mean by high frequency, and why is that important for you?
Thank you for the question. Thank you for the question, Andreas. Yes, high frequency recording means that I need a time series with many data points. For instance, if I take EMG data, I can have, let's say, frequency recordings about 1000 hertz. This means that if I take EMG data and I collect EMG data during 10 seconds, since the sampling frequency will be about 5000 hertz, I will obtain 10,000 data points. This is very important. Why it is important? Because unfortunately, our data analysis methodologies are data length sensitive. So we need long time series, very long, in order to apply this time delay stability or these other spectral or frequency analysis methodologies. Otherwise, it will not work. We need a long time series so we can identify couplings, and therefore we need high frequency recordings, at least 500 hertz, which means 500 data points for each second.
And just to give an example of that, when you collect VO2 max data, you know that we have the option to go minute by minute or raw data or second by second or breath by breath. So in that case, we prefer to go breath by breath. So we have to get all the data, right? So for the rows that I saw you over there, because we applied the novel also a rowing ergometer, because we're actually discussing to implement something with the USA rowing team, we changed the protocol. So we had intermediate protocol because this is how the rowing as a sport they're doing. This is how they test in the real field their athletes. So our analysis, okay, that's some that we have in the manuscript, is that maybe it's not the best because we didn't actually apply the basic rule of this network physiology of exercise. That means that we have to have raw data in a time series because of the protocol. The beauty of this right now is that because I'm collecting, again, a second set of data for these rowers, I can compare at least and see with the same protocol we did, how they change. And FYI, our team won the national title in Division 2 rowing. So that would be great for me to see, okay, how the training maybe was part of this story and how they changed pre and post.
I have a follow-up question, and then I'll let everybody, because this is really interesting to me, obviously. You don't necessarily need specific variables. Your innovation is how you use a knife to cut it. So you cut it differently than other people. So you can take the same data set that I have and somebody else and just analyze it differently and show me that coordination between systems. And I can be a very good rower, but not have a good coordination.
Correct.
And I can be not a good rower and have a good coordination, which is, so you compare it in terms of if I reach my potential or not. So it's between the individual, if I understand that correctly. So then your end goal, let's say you figured it out. And we know at any point of time, in any sports setting, in any team, who has good coordination and who has bad coordination now, because that changes, right? Something can happen. What's your end goal to create interventions to fix that and bring like a bad coordination to good coordination, or because that sounds like a long project. That's a huge journey, which is exciting. I'm just trying to see where are you going.
The area is brand new. It's like how many years, like in the field?
Network physiology per se was introduced, as I said, by Professor Ivanov 10 years ago, more or less. So, and we start applying network physiology concepts to exercise maybe a couple of years ago, 3 years ago. So, I mean, your question is great and your point is great, and thank you for making it, Andreas. Actually, that's true. I mean, we can analyze, we do it. We analyze databases utilizing different techniques, and we obtain different information. That's great, and that's the real point. We don't say that our methodologies are better. We can say that with our methodologies, we can complement, we can offer unique information that can complement the information provided by the commonly utilized physiological markers. So that's the point. You can put it together. So, and then as for the last part of the question, yes, I mean, our final aim would be to, okay, now we need to understand how different organs and muscles coordinate. So we need to understand this first. So once we have all these biomarkers, and we hope to have them, develop all these things in the near future. So then the next step will be, okay, how can I train? What should I do to improve coordination? That's the long-term aim. Okay, or how can we utilize this coordination to prevent injury?
Just to get an idea, the Keck laboratory in Boston doesn't collect any data; they analyze the data. So we want like researchers to have ideas, okay, share the data with us, and then as I show you with my study, the one that I did with traditional like perspective, and then to reanalyze with this one, because we try to create normative data. We try to see if that new concept actually gives a value. We don't know if that gives a value, but we see that there's a promise over here that, you know what, the coordination might be something that we need to train, because we know, let's say for based on the ACSM guidelines, you need to do, let's say, basic strength, 8 to 12 reps, okay? What about 8 to 12 reps doesn't give you that coordination? Maybe the trainers need to get something different, okay? Different numbers, different like number of sets, different like, let's say, I don't know, break between the sets to improve that coordination. Because what was clear with the data from the rowers is that the Olympian, okay, didn't have the highest VO2 max, okay, and didn't have the highest coordination. And I had other like athletes that they didn't make the All-American team. They're actually playing other sports, because in Division 2 we share scholarship between other sports. She was actually trying to make the team, to help the team to make the numbers, and I think it was a basketball player and had the best coordination. So maybe that can go to the cross training between sports. You know what? We have kids start with gymnastics, start with swimming, and then move to track, then move to other sports, because you get skills that relate to maybe coordination. And then goal is to get all these algorithms to put them like in maybe in a wearable device, and then try to see to get a feedback, to get like a point of get up, walk, do something like this.
All right, thank you so much for the presentation. I thought this was actually extremely interesting. I hadn't heard of this before. I'm a little curious. It's kind of a two-part question, but however you guys want to approach it. So there's going to be variables based on the need for that, I guess you could say, high data yield that you can't actually analyze with this method. So like blood work, for example, you give an example of we don't have that high frequency yield. So then also, because you don't have those factors, are we not able to get the full picture? So you mentioned the rower who had not the highest VO2, not the best coordination, but maybe the highest percentage of her VO2 before she reached her anaerobic threshold. But you couldn't measure that without maybe a blood marker. You could, predict it, but how does that kind of come into play here with coordination?
You want to take the first part with the coordination of the lactate threshold, and I'll take the other one.
Okay. So, I mean, the question is, how can we quantify coordination if we don't have long frequency recordings?
That's the first part. Yeah.
So the point is, right now, we need high frequency recordings. because I mean, at some point we can utilize, for instance, with the, the paper that Zach presented with sleep deprivation, we didn't utilize high frequency recordings, but then we use like simple correlations. Simple correlations, of course, can provide information and it's good, but it's not the real essence of network physiology. So for now, we really need time series, long time series. Because this way, we can have information about the dynamics of the physiological variables and how these organs dynamically coordinate. This is of key importance because the dynamics provides you very important information, okay, about how this physiological signal evolves in time. Therefore, I mean, right now we need long time series. For cardiorespiratory data, we need breath by breath data. Maybe in the near future, we will be able to develop new algorithms that can allow us to, to identify real couplings with long, times with short time series. Sorry, but for the moment, we need high frequency recording. That's the limitation of technology.
So to go to the other part with the blood that you said, yeah, that's a weakness. That's definitely a weakness. But I'm still like part of the science that I'm saying that we have to intervene with whatever we do. I don't believe completely to the artificial intelligence. Dr. Bolani said that, oh, we need to reach a point that we're going to have a device to make decisions for us. No, I need an individual to do the exercise prescription, to be able to get information, to filter that information and give me the perfect one. And yes, with the blood work, okay, I'm not getting the data that I need in time series, but I'm going to get some indication. Actually, Dr. Bolani said about fatigue, right, and it's about emotion. And I was able to hear like a speech of Dr. Inoka from University of Colorado in Boulder. He's a great like neurophysiologist, and he studies fatigue. And he said that it took him like 30 years to understand that fatigue, okay, it's an emotional feeling from the efferent like system that gets like to the respect, like let's say brain centers, how we feel. And it took him 30 years. He's like like science about that. So yes, there are like different ways to see a problem, and this is what we try to do: try to provide more information, biomarkers with limitations, right? So it's about the laws, okay? For me, it's very strong to say that we try to create like universal laws because it's a brand new area right now. That's why we seek collaborators. That's why we seek like people that they want to emerge in this, like, let's say, new field and give us access data. We can get the same concept and we can do like a reliability analysis at this somehow. We take the same design, we analyze different and see if the results, okay, the outcome is the same. As with my study, no, it wasn't the same. That it's a point of direction that we can follow to think out of the box.
When speaking clearly, coordination variables are able to provide information that typically recorded physiological variables are not able to provide. That's the key. That's for sure, and that's why we need to complement. That's very important.
Based on that, actually, if you don't mind, one more question: is it possible that I think the purpose is kind of to characterize the most efficient coordination, that we could have two athletes in the same sport that approach efficient coordination differently? So I don't know, let's say a 10K runner who's a great finisher versus the pace setter. Their optimal efficiency would be different, one versus the other, and you guys might be able to characterize that.
Correct. This is something we can do because we say that we may have the same two runners that they have the same VO2 max, but who's going to win the race, right? The one that has the highest electric threshold. So that's a level of extra information you can give to your coach or to your athlete. Pretty much the same with the coordination. You may have the same elements, identical twins, but you coordinate differently. You respond to exercise differently. Okay, this concept may be able to give that, let's say, science behind the responders and non-responders in the exercise.
Anything else?
Can you take it up?
Oh yeah. The next step is
the question
in clinical population.
That's the question for Zoom because some people. So I asked if you can take it to general population and if it's something that you can be applied to pretty much everybody.
The point is to generate. We are first working. He presented data about elite rowers, but we are mainly working on healthy people because we want to know how this coordination looks like on healthy population. For example, like
sorry. For example, in cancer patients, we know that what we see consistently in all types of cancer is that on the micro level, the cell itself does not communicate correctly. So if coordination is a big thing, like the application can be, which is a big thing. I believe in it. I cannot prove it because you start doing it right now, but I know everything coordinates, obviously. But I feel like the application can be everywhere.
Absolutely, Andres, and this opens, huge and exciting horizons. That's great. Actually, yeah, we are mainly working on healthy, but we have, also some works on Parkinson and Down syndrome, and we are finding that, of course, coordination is different. And a network here, we showed you some networks. A network can break down because the links disappear or because links become stronger. So we are finding that in neurodegenerative disorders such as Parkinson or even Down syndrome, the networks become very rigid. The links becomes very strong. This means that the network, the neuromuscular system, becomes rigid and loses adaptability and cannot respond to the different environmental constraints. So we are on it, and yeah, absolutely, this has applications for clinical populations. I want to go step by step and first, build a solid basis for healthy people, and then we will go to clinical, more elite athletes, different pathologies. But yes, absolutely.
Imagine, I think that we have the so-called metabolic syndrome, like a risk factors, and we have these 3 out of 5. You have metabolic risk. You have metabolic syndrome. What if we take it, the constant, we say, hey, what about the coordination? Because you may have, let's say, high lipids, but you exercise or you like a low blood pressure, that actually interacts, coordinates different, and then the outcome is to be healthier than somebody else based on this like rigidity of the network that we can present. And that's why I want to establish the baseline on the health population. We have health population, athletic population, and then we can do the same with clinical population with different diseases to see how this pattern, how this rigidity of the network changes.
We're good.
Thank you.
Thank you.